So to remind you all from class the other day, we talked about cell theory. And cell theory has 3 components. So all living things are composed of cells. So living things composed of cells. And then that cells are the basic units of structure and function in living things. And then that new cells come from existing cells. So I want to talk a little bit about this last one, that new cells come from existing cells. And if new cells come from existing cells, then where did we get the first cells? So if cells are the basic unit of life, then if we talk about the first cells, we're talking a lot about the origin of life. And the origin of life is also called abiogenesis. So the prevailing hypothesis about abiogenesis is that going from non-living things, so life arising from non-living matter, such as simple organic compounds. So some of these organic compounds are the macromolecules that we talked about. So those macromolecules are, again, lipids, nucleic acids, proteins, and carbohydrates. So this process of going from these non-living organic compounds to actually fully functioning cells. And the prevailing view is that this wasn't just a single event, but it was a process of increasing complexity, so multiple steps. So early Earth looked something like this. The planet's formation was from this solar nebula and a process that you guys don't really need to worry about, but there was a lot of intense meteorite bombardment, including all these big impacts with the Earth that created a lot of this like magma across the Earth's surface. And that led to the atmosphere of the Earth being something like this. So as you can see, no oxygen, but there was some methane, CO2, this is ammonia, water vapor, CO2 again, nitrogen, sulfur dioxide, etc. So how do we go from this pretty extreme atmosphere with no oxygen and no living things, and not even any real large organic molecules, to cells? So in 1952, Miller and Urey ran an experiment where they simulated the conditions of this early Earth atmosphere in a series of experiments. And their experiments looked something like this. So the water vapor that existed in the early atmosphere did condense and form small pools on the surface as the Earth cooled. So that's where this water right here is coming from. So Miller and Urey replicated this early atmosphere by inserting these gases right here, like methane and ammonia. And so this water vapor here, and then they inserted these gases, and then those gases were set under a spark, so some electricity, because there was a lot of lightning going on in the early atmosphere. And so here you can see early atmosphere gases. Here's what's supposed to be a lightning, the spark. And when those gases were subjected to that lightning and then cooled and condensed, they looked back at this water here and they found some really interesting stuff. And in the first set of experiments, what they found in the condensed water were 5 amino acids. And if you guys remember, amino acids are the monomers, the building blocks for proteins. This first experiment inspired many others, and by manipulating amounts of the gases and some other stuff, they actually were able to get many other organic molecules being formed in similar ways. So one theory is that life originated at hydrothermal vents. If you remember hydrothermal vents, we talked about them a little bit in terms of chemosynthesis, right? So they're like the one example of where the sun isn't used to produce energy through photosynthesis, it's chemosynthesis through chemical energy. So you have a lot of those chemicals being produced at hydrothermal vents that were used in the Miller-Urey experiments, and you have all of that water that's sort of the medium for this creation. So if you have the production of amino acids, that results in proteins, and proteins carry out really important functions in cells, but in order to have a cell, you need some kind of barrier. And by manipulation of the types of ingredients in these experiments, they were able to get lipids as well. And if you remember lipids, so if you remember our discussion about lipids, lipids have a hydrophilic head and two hydrophobic tails. So this part is hydrophilic, meaning water loving, and this part is hydrophobic, meaning water fearing. If you remember from our discussion about how phospholipids are used in cell membranes, the hydrophobic tails want to be away from water and the hydrophilic heads want to be near the water. So this double membrane forms, this bilayer forms where you have two phospholipids facing each other so that the hydrophobic tails are inside the membrane and the hydrophilic heads are on the outside of the membrane. So if you get a whole bunch of lipids all together, they're going to self-orient in this way so that if you get a lot of them all at once, you get something that looks like this. So some of the first cells were something called prokaryotic cells. And the cells that you have in your body are eukaryotic cells. So we're going to talk about the difference between prokaryotic cells and eukaryotic cells. So just to give you an idea of what the difference is and how they look, prokaryotic looks like this, and eukaryotic cells look like this. So there are some pretty big differences between prokaryotic and eukaryotic cells. I'm going to outline those down here. So for prokaryotic cells, they have a singular DNA molecule that's circular. So the DNA is not organized in chromosomes. They have no nucleus. So the nucleus is a membrane-bound capsule that encloses the DNA, but they don't have one. And they don't have any specialized organelles. So organelles are compartments within eukaryotic cells that perform specific functions. And they only have one copy of each gene. So in contrast, eukaryotic cells have DNA that is arranged in chromosomes. And that DNA is enclosed in a membrane-bound nucleus. And actually that nucleus is right here. And eukaryotic cells do have membrane-bound organelles. So some of those membrane-bound organelles are like right here. This is a mitochondria. Endoplasmic reticulum is right here. Golgi apparatus is right here. And we'll talk about each of those different organelles in particular, like individually. And then eukaryotic cells also have two copies of each gene. So you get one copy from your mom and one copy from your dad. So prokaryotic cells are mostly bacteria. Eukaryotic cells are plants, animals, fungi, etc. Some things that prokaryotic and eukaryotic cells have in common are they both have this plasma membrane. That plasma membrane, again, is made up of that phospholipid bilayer. They both have these things called ribosomes. Ribosomes are these really tiny non-membrane bound organelles that assist in making proteins. And prokaryotic cells have this cell wall, and plant eukaryotic cells have a cell wall, but the cell wall is made up of different stuff, so it's not the same kind of cell wall. And then they both have cytoplasm. So cytoplasm is the fluid that makes up the bulk of the cell. So prokaryotic cells were our first cells. Bacteria were the first living organisms to be found on early Earth. And then eukaryotic cells came after. And the theory of how we went from, how we evolved from prokaryotic cells to eukaryotic cells is something called endosymbiotic theory. It looks something like this. So here's our ancestral prokaryote. So you can see it has DNA and cytoplasm, but it's pretty simplistic in terms of a cell. It has this plasma membrane. And then if you remember our eukaryotic cell, I'll give you a little reminder. So here's our eukaryotic cell, and on our eukaryotic cell, we have some of those organelles that are just a lot of membranes, like right here, that are folded in on themselves. So here's one, this endoplasmic reticulum. The Golgi apparatus is another one where all that membrane is just folded in on itself. So I'm going to shrink this down for a minute. Put it over here. Right. So in this, you can see right here that the first step was you started to get that plasma membrane folding in on itself and creating some of those membrane-bound organelles, the Golgi apparatus, the endoplasmic reticulum, which is just the membrane folded in on itself. Right. You also started to get the development of this nuclear envelope in the same kind of way that enclosed the nucleus and enclosed the DNA. Then here's where it gets pretty interesting. Here you have a large prokaryotic cell actually engulfing these smaller prokaryotic cells right here and not digesting them, but actually incorporating those smaller prokaryotic cells into this larger prokaryotic cells. So those were like the first mitochondria, and we'll talk about mitochondria more and how they help produce energy for the cell. If it's a photosynthetic prokaryote, it would have engulfed these smaller prokaryotes called chloroplasts, and then those smaller prokaryotes, they are actually double membrane bound. So if we go back to our cell right here and we enlarge it, you can see that the mitochondria actually have a double membrane. So they have one membrane on the inside and one membrane on the outside. And the theory is that happened because when they were engulfed, right here, that membrane that assisted in engulfing them actually became that outer membrane of the organelle. So then you start to see these smaller prokaryotic cells being engulfed by the larger prokaryotic cell, like the mitochondria and the chloroplast, and then those don't get digested. They actually start to perform functions within this large prokaryotic cell, and then that large prokaryotic cell eventually becomes this first eukaryotic cell, which would be right here. So again, if we go back to this eukaryotic cell, you can see that it's different from the prokaryotic cell because it has this nucleus right here that is membrane bound and it's closing the DNA, which is these chromosomes. It has all these infolding membranes, which make up the endoplasmic reticulum right here and right here, or the Golgi apparatus. And then it has all these special membrane-bound organelles, like the mitochondria and plant cells, the chloroplasts, that started to perform various functions for the cell, and we'll talk more about that later.